So I worked predominantly in Norway for the last 4.5 years. And what I was doing was working directly with beekeepers in a resistance breeding program to kind of replicate the methods of one particular beekeeper who had actually bred resistant bees before. So what we were doing this time was we were doing similar things to what he was doing, and then I was taking a bunch of data so that I could see how the populations changed. So that's what I'm going to be presenting to you now is like an active breeding program and what we got in about 5 years. So that was a pretty long time for research, which is why I was excited to take the project. So I'll give you a little bit of a crash course on Varroa. I know we covered a lot about the basics of Varroa and Varroa reproduction and stuff in other talks, but I've got a little more to add and some cool data to share from other papers. I'll talk about breeding for Varroa resistance in Norway, predominantly where I worked. What I found there, the patterns, the behaviors, and what we took away from that to add to this new breeding program that we just did. And then I'm going to share some of the results from the four. I said 4 years. There is actually 5, but we're missing some data. So I said 4. I called it 4 in the talk, and I'll show you kind of what's happened, what we've seen, what worked, what didn't work, and yeah, we'll start on there. So we know Varroa. This is a juvenile. This is a pseudonym for female Varroa here in the picture. That's why it's white and see-through. It's an invasive parasite, comes from Asia. It's usually on Apis cerana, which has defense mechanisms, so they can't reproduce in worker brood. They can reproduce in worker brood in Western or European honeybees, though, and they do, which causes the exponential growth curve of Varroa, which allows them to get out of hand so quickly. You've got elevated colony losses due to the mites, and because of the viruses, it's spread. So virus is adapted to use the mite as a vector. Titers just went through the roof, so the levels of viruses in the bees, and that was what was killing your colonies. I think Dr. Trainer illustrated it very well with that really cool video we got to see. The other bad thing about Varroa is it tends to adapt to chemical treatments. If the same treatment is used again and again and again over the course of years, a lot of the mites actually start adapting to it, which means that a lot of the chemical treatments are now probably not long-term viable options to control the mite. So this is why I was kind of starting to get interested in resistance breeding, is because what natural resistance does, it actually keeps up with the evolution of the mite. The bees are changing with the mites, so that you have something called this dynamic equilibrium, where usually things stay in kind of a tentative balance with each other, and it's hard to knock it out of place. So you got a strategy then that keeps up with the changes of the Varroa, and prevents it from adapting too quickly, so that it gets out of hand again. We now know that there are a lot of populations of Western honeybee, of European honeybees that can survive Varroa without any sort of treatment at all. There are the populations in Europe that I've studied, the Norwegian population. There's actually 2 of them at least there. There's 2 of them in France that I know of. We've got the bond bees in Sweden. Professor Stephen Martin, who's been working with me on a couple of projects, he went to Cuba and researched the bees there. They didn't have the means to treat extensively in Cuba, so they didn't. So when they got the mites, they lost a lot of colonies and they bred from the survivors, and now almost the entire island of Cuba is treatment free, completely Varroa resistant. He's also got populations in the UK, and I know of several others that I won't get into, because if I sit here listing the number of populations I know of, we'll be out of time in no time. So some things you might not have known about Varroa. They've got a chemical mimicry strategy. So in about 4 to 6 hours of entering a hive, they actually smell exactly like that hive. So they become almost invisible to detection unless they're actually jumping on bees. So it's very difficult for the bees to locate them. The foundress does take proteins from the bees when she feeds on them, and she uses that in egg production. They are fully aerodoticous, which is basically the English version of that is they are haplodiploid. They can lay haploid eggs, so eggs that come from basically just has one set of chromosomes. So unmated females can actually lay male eggs. That's something they can do. So they don't need to be mated to produce offspring. And that's important if you're looking at population control. So even if mating is interrupted, you've got a foundress that can go into another cell, lay a male for a completely fertile foundress, and spread genes that way as well. The males produce a very conservative amount of sperm, so they really only produce as much as they're going to use. So if any of that is wasted at any point in time, they don't have enough to complete a fully reproductive cycle as well. So a little bit more about the what they feed on. So they feed on the fat body tissue, not the hemolymph. And that was found out by this really cool study with the reference down at the bottom there, where they stained hemolymph in yellow and they stained the fat body tissue in red in a bee, and then they let the Varroa feed on them and then looked at what stain ended up in the mite. And it was mostly red, so it was the fat body. Another piece of evidence is to how that it's the fat body and not hemolymph is there's a very localized feeding site on the bee that the mite almost always go to. So they almost always go to the left side of the bee in the secondary tergite there in the abdomen underneath that little red dot in this picture here. So yeah, that's important because the fat body again helps with overwintering. It's involved, the vitellogena is involved in immune function, and so that is much more important to the bee than a bit of hemolymph. So the mites are actually taking very important resources from the bees when they bite and feed. In terms of the viruses that it spreads, a very interesting story with deformed wing virus is that not all strains of deformed wing virus actually vectors with the mite. There was a really cool study done by Stephen again, looking at viral diversity in Hawaii on the islands where Varroa had progressively invaded. So the different islands was kind of like a timestamp progressively as how many years Varroa had been there. If you take a look at the colored circles at the bottom there, the island of Kauai had basically no mites at all at that point, and the viral diversity was very high. But as soon as you start getting Varroa and like even 1 to 2 to 3 years, that viral diversity drops off, and you get basically one strain. And that one strain was the one that was able to use the mite as a vector and spread. And we know it kills colonies because we've done studies, and there was more recent evidence presented beforehand. But I'll present this study, which was done in 2012, where they were looking at the number of viral copies that these colonies had, and the bees in the colonies had, and then the number of Varroa, and then whether that colony lived or died in over that winter. And we found that the colonies that died had much higher Varroa, but they also had much higher viral loads as well. So it was pretty good evidence to illustrate that viruses do play a very large role in killing colonies. Right. So my story began in Norway when I started my PhD, where we'd heard tell of a beekeeper who had not treated his bees since about 1998. It was about 2013, 2014 when I started the research, and he was keeping his bees just fine, and he wasn't using any sort of chemical treatments. The only treatments allowed in Norway were formic acid and oxalic acid. I'll say that right now. So we actually didn't use synthetics to treat for mites there. But what Taria did was, the the original instructions on treating were based on regions much further south, and they were actually much higher levels of treatment than the bees in Norway really needed or could tolerate. So we ended up losing a lot of bees at that time as well. So at that point, Tadia said, either I can kill my bees with this treatment or I can let the mites kill them. I choose, I'm going to stop killing my bees myself. And he lost almost all of his stock. So I'll say that right now. This was not an easy thing to do. But he bred from the survivors. And in about 5, 6, 7 years, he had replaced his stock almost completely and was keeping a very competitive honey operation without treating in any way, shape, or form. So that's where I came in at that point in about 2014. I was sent, they're like, go over there and find out what it is, if they're actually surviving, what they're doing. and that was my PhD. So I started monitoring, and uh, pretty much every single time I went out there in the summer to count mites and look at them, Taria's bees, which are going to be illustrated in blue, they're the blue lines on these graphs, and the control populations that were regularly treated, they were in orange. Taria's bees always had lower mite counts, and these were bottom board counts. And there's the average daily mite drop. The number of colonies are outlined by the numbers on top of each bar. Um, and there was only one time where I didn't find a significant difference, but they were still lower. Was probably when I didn't have enough colonies to run statistical tests. But almost every other case, the mite counts were lower. And most of the time, I was taking data in August when the mites were generally getting out of control, when people were treating. Um, so I mean, he wasn't treating, and they were that low. So I decided to do a little more intensive study when I finished my PhD, and I got this project working on for the last 5 years in actually in Norway. I was living and working in Norway running this Varroa project. Um, the first year I was there, I decided to put bottom boards down every single week for the entire season in about 25 of Taria's hives and 25 control hives to monitor the mite fall through an entire season. And this is what I found. So the first week started around mid-May. And the last week was about mid-August. And I'd say actually it was supposed to run for 15 weeks. And I had to stop it at 12 because I was so afraid of the control colonies all dying before I could collect meaningful data that I decided to stop the experiment and treat them. And we still lost a number of colonies. But Chinese colonies were fine. They were they were absolutely fine. Um, the beginning of the blue line here and the end of the blue line weren't actually statistically significant, even though it looks like it raced a little bit higher. The beginning of the orange line and the end of the orange line, that was definitely significant. And of course, the average drop by mid-August was very, very different between Taria's colonies, these survivors, and the control population. So that's pretty convincing for me is that these bees had the ability to just control Varroa population without being hindered. This is another breakdown of that. So this is at the time point of August. This is the number of colonies, the percent of the population that had between such and such number of mites. And again, Tari is in blue, and about 80% of his hives had between 0 and 10 mites on the board, which seems very high. I know the cutoff being 10, but this is these colonies surviving in Norway. Norway has a much lower colony density, which I think might be playing a role into why the mites can be a little bit higher. But these are the numbers. Right. So, and we can see the control population actually had a huge spread. Not a lot of them, but some of them were actually between 0 and 10 in August, which may represent that you actually did have some colonies with a high potential for Varroa resistance in the population in the mix, but you also had a lot of these mite bombs, which were probably driving the numbers up as well and making it harder to see the colonies that did have potential. Right. So I ran an experiment. This was during my PhD, and this is the pretty much the only time I have found very significant differences in the reproductive rate of Varroa destructor when I was looking at the brood in colonies. And what I had done was instead of just taking the natural reproductive rates, I took brood frames that had been freshly capped from donor colonies in a completely different region. And I took, I think, about 20 different frames, and I took them and split them and put some in Taria's colonies and some in the controls, and let them basically ride out their capping period in those hives. What I was testing for was worker hygienic behavior. I wanted to see if you had a natural Varroa trigger of this highly infested brood. Would there be a difference between the reaction of Taria's workers versus the reaction of the control workers? So we kept them in for about 9 days post capping, which is the best time to assess mite families because they're all at different stages and you can read them. Before we put them in, we actually did take quite detailed maps, brood maps, and photographs. And then individually, I dissected each cell, and every infested cell, I counted the mite families and recorded the reproductive rate. So the average reproductive rate of Varroa on a susceptible colony, on a colony that needs to be treated, is about 1.34. So our controls were very close to that, 1.24. But Taudia's colonies had a mite reproductive rate of 0.87, which is below 1 foundress per, or offspring per foundress, which is very low. And so we had the low number of mites, and then we had evidence that, just for the capping period, these workers could affect the brood enough that they could reduce the reproductive rate of these mites. And I'll make another interesting point is I also tested for how many of the cells were actively removed with these maps and photographs, and then opening each cell to make sure another egg hadn't been put in and the cell had been capped again. And the level of VSH, or level of brood removal, I found in this experiment, they were not different between the surviving colonies and the susceptible colonies. So there was no elevated brood removal in the August time point that I collected, which I found was very interesting because we found brood removal in pretty much every other population that has developed resistances. So equipped with that information, we were going to start what was called the Herdal breeding program. And what this was basically, it was a queen breeder who had followed Taria's work and had actually stopped treating before we started this one queen breeder, and wanted to do this breeding program. And the Norwegian Beekeepers Association got funding to actually investigate this, thanks to Taria's work breeding his bees. So we got this program, and then they offered it to me to design and basically make the selection algorithm and guide, what queens got to be bred and how the whole operation worked. So I'll introduce you to one of my favorite queens. She's kind of become the mascot of the program. This is Blue 42. And it's completely by fluke that I got a picture of her. Because I marked all these queens individually, and she ended up being one of the best performers and actually produced really good queens as well. Not all of her daughters were fantastic, but a lot of them actually had very high levels of hygienic behavior and very low mite rates, mite loads in the critical period. So the whole overreaching goal of this project was to create a method that beekeepers could use, a reliable method to select Varroa resistance in their stock. So these were using methods that were, easy to do, that were not time consuming, that were not expensive. And then you had me in the background taking all kinds of data to see if this simple method worked. So we had some things that really made it work. First of all, we had a whole community of very interested beekeepers involved and willing to help. They took data the way that I trained them, and they gave it to me with very little compensation. We had a local queen breeder who was very enthusiastic. That's him on the bottom right. That's Ruar, and Taria is on the left-hand side there. They're grafting queens in the picture, and I'm standing in the background. You can see me. So we had Ruar on our team. We also had a fairly isolated beekeeping area. So we had this valley with a lake in the center, and a very low density of colonies on the outside of this valley. And most of the beekeepers in the valley were part of this program. And what they did was basically they agreed to buy queens from our local queen breeder exclusively, and rear them, and then supply the project with data so that we had about 100 colonies in any given year that we had data on. Right. And then so we also had, from Taria's work, we had a kind of a profile of what a surviving population, what a Varroa-resistant population looked like in Norway. And the project has been going for 5 years. I'm actually not working directly in Norway at the moment, but I am still working with the beekeepers. I'm still going to give them their data sheets in spring, and I'm going to help them through the data collection and analyze it at the end of the year. This is something that I chose to do because I think it's incredibly interesting, and I want to help this program succeed. It's actually now started in a couple of other areas in Norway too, completely organized by the beekeepers themselves, using the algorithm and the methods that we developed. Right. So what sort of data do we collect? So as I said, we had just over 100 colonies each year. which is a really nice sized population to take data from. And I asked them to take data 3 times a year, plus the honey collection. So it was in spring inspection, it was when the first supers went on the hives, and then when the last supers were taken off the hives. Those were the 3 data points. And the way I had that instead of specific points in time of the year is because the climate is very different year to year. It can be volatile, and the bees will respond to the climate, not to the timing. So we took bottom boards, and I realized that's not the most accurate method, but talking with the beekeepers, knowing what the weather was like in Norway, that you could have 2, 3 weeks of rain sometimes, you had cold snaps. They were not too enthused about opening their hives to take mite washes. They didn't want to kill their bees. So bottom boards was something that they were familiar with, something they were happy to do in whatever weather. So we went with those to kind of get a general population count of the mites. We also had metrics like colony size and status. We had general health scores. We had gentleness scores. Because it was very important that we keep up these commercially desirable traits in the breeding program and not lose them while we were also selecting for Varroa resistance. And we also had queen ID, and every single queen in the project had a tag and was registered so that we knew exactly where she came from. And then in addition to all of that, I went and sampled 10 random colonies in the entire population each year and took brood samples in August so that I could run, my reproductive success. I could run recapping studies, and I could basically look at all the stuff that we couldn't see when we were just selecting for bottom board counts to see if there was changes as we went. So this is what we started with, and this is I threw this the Herdal bees up on the graph that I showed you before of the number of colonies that had between 0 and 10 mites and 10 and 20 mites in the critical period of August. And we can see that the queen breeder who supplied most of the colonies for this assessment, the the colonies were about halfway there. So we had about 50% of the population between 0 and 10 already, which is kind of the gold standard. But we still had that tail of mite bombs that we really wanted to get rid of. So that's what we started with. Yeah. So the results. So we were breeding for about 4.5, 5 years, and then progressively, these are the population, the mite drops for spring, summer, and autumn. Those three points that I told you about, and how they changed over the years. So first, I'm going to say is 2020 was actually a golden year. It was so nice and so warm. There was so much honey, and all the bees were happy and healthy. There was hardly any mites. It was a great year. And then over that winter, we had about twice the national average loss rates in the entire area. And a lot of bees died everywhere, not just this project. So we had, after that disaster winter, we had very high mite rates and low honey production, and it was just frustrating. But if you're taking a look, so going from light to dark, and then blue is the final year, just last year, we actually do see that there is a decline, especially between 2022 and then 23 and 24. We see a decline in the line, but only in summer, which is very interesting. And I really wanted to know why the mites were shooting up back up in autumn, even though you seem to have something not dissimilar to Taria's population control for most of the season. So that was interesting. We're going to talk a little bit more about why that might be happening and stuff. But the fact that they kept it low for the summer was really, really interesting. And I think one of my main theory now is that because there was absolutely no mite management in the project when we were doing it, it was only selection, is that maybe if we had done a bit of mite management from summer to autumn to help the bees, we might not be masking our survivors in the mite bombs that were definitely mixed up in there. So brood infestation rate, and this is the most interesting result to me. So I was looking at the number of mites in these 10 frames that I that I sampled in August randomly throughout the population and taking kind of the average number of mites that I'd found in the brew. So how many cells were infested, how many cells weren't infested. And 2010, we had a 15% infestation rate as expected because this was like a great year. And in 2021, that shot up to about like 27% brood infestation rate. So 27% of the cells I dissected had mites in them. And then 2022, we actually went back down to 15, which was nice. But the interesting thing happens then because we have a progressive decline. And in 2023, it was the lowest I'd ever seen it. It was about a third of what we started with in 2020. So there were almost no mites in the brood in the critical period in August, which I thought was very, very important. Because if you don't have any mites biting your bees in autumn, then your winter bees theoretically are going to be virus free and relatively fine. So that was an interesting result, right? So now I'm going to talk about this recapping behavior, and why I was harping on it so much is when I was looking at the first study where I introduced the infested brood to Tarius colonies and then to control colonies to test the worker ability to control mites. I didn't find any brood removal, but what I found was this behavior where the bees were chewing open the cell caps and then capping it back over. And I found it in droves, like almost every single cell had this recapping performed on it in the donor colonies that I was in the donor frames that I was using. Um, so no brood removal, but the mass amounts of this recapping, just every single cell. To test this further, when I pulled these results off and wrote about them in my PhD, I went to France and Sweden, who I know had populations of bees that we had data of Varroa resistance. And I was like, hey guys, can you test for this? Because this is a really interesting finding here in Norway. I want to see if it's present in other populations as well. And sure enough, it was. So the graph at the bottom is fairly complex, but basically the dotted lines divide each of the populations. And then you've got these two bar categories of whether the cells we dissected were infested with mites or where they weren't infested with mites. The horizontal axis here is the proportion of recapped cells you found in each of these categories. And then the dark lines are colonies that had resistances to mites, and then the light gray bars are the colonies that were susceptible, that had to be regularly treated. So right away you can see that the highest bars, so the highest rates of recapping were in surviving colonies in the infested cells. And these populations had nothing to do with each other genetically. They weren't linked in any way. So the bees therefore had basically developed these high levels of recapping completely on their own at the same time, which is really solid evidence to say that this is definitely something that is associated with Varroa resistance and high hygienic behavior. And other researchers started to look for it as well. Stephen Martin was one of them, and found it again in Brazil in the Africanized bees, and also found it in Cuba. So we have this trait now that's very highly associated with Varroa resistance. Can we use it? So there's a couple of reasons for variation in this trait that you're going to see. There's two different elements to this trait. I kind of divided it into, there's the frequency of recapping. So how often how often the behavior is performed. And then you have the accuracy of the recapping. So how often the bees get cells that actually have mites in with this behavior, because if you're missing all the time, it's wasted energy, right? And I found that when I analyzed these colonies, there were actually variations in these two different elements of the trait. So some colonies recap things a lot, but they missed a lot of the mites. Some didn't recap a lot, but when they did recap, they actually got the important cells almost all of the time. And then some colonies were great at both. They recapped a lot, and they also had very high accuracy. So there is a bit of a trick to this, and some of the other talks brushed on it, is if you're assessing a hygienic trait, the levels of this expression vary with the level of threat in the colony. So if you have a colony that is a really good survivor and it doesn't have a lot of mites, you might not see the behavior that would get it there, right? Because there's no challenge. Which is why hindsight thinking as to the very first thing I did when I introduced these colonies, or these high mite-infested frames into these colonies, I actually provided the challenge, and I think I managed to trigger the behavior. At the same time, if you have a colony that has a high mite load, you want to know why, right? So high enough that you can observe the behavior. Well, why are the mites high? So this is maybe what makes it not the greatest trait to actually, use for active selection is because the variability and the presence of this trait is so high, the variance of this trait. So how often it's expressed, when it's expressed, in what level. So yeah, false negatives is definitely what you're going to see. Um, so, I mean, how useful is it? I would say the presence of recapping is definitely going to clue you into something that might have a high potential for survivability. But as for an exact measure, it may not be great unless you had a trigger. UBO, for example, maybe, I'm not sure. Right. So again, to illustrate, so other studies were done looking at recapping. It was present in pretty much every single population that had resistances to mites that we cataloged. I think to this day, I don't think I don't think we have not found it where we've looked for it in resistant stocks. So again, very highly associated. Now let's talk about the change in recapping accuracy. So the success of this trait. So how well it was performed, how often they got infested cells with this. And really interestingly, it was very low in 2020, and it actually just kept getting higher. And the percent numbers on top of each of these bars are actually, it's not the level of recapping accuracy, but it is the percent infestation rate in the brood. And why I did that is to look at, even though the infestation rate, the challenge is decreasing, the level of accuracy stays the same. And this is actually from 2020 to 2022 and 3. That's a significant difference. So the recapping accuracy was vastly improved in the population over those 4 years of selection. And so it's very interesting. We actually actively affected those behaviors. They changed because of the selection program that we put in. So this slide, a lot of information is basically just to summarize everything that I said. So we had these 4 years where I was looking at brood infestation, the number of recapped cells, and the recapping accuracy. We didn't manage to get samples for 2024, unfortunately, but we have these 4 years of very solid brood data. And we're looking at the percent of brood infestation absolutely tanked in 2023. Which is really, really interesting when you compare it to 2020 and 2021. And then on the right-hand side of this graph, you can see how much of the population was replaced with the stock that we selected for. So 2020, we had no queens that we had actively selected. In 2021, we took the data from 2020 to actually advise the selection. And then we replaced about 50% of the stock, give or take, out of those 100 colonies, 50 queens were replaced. And then 2022, we had about 83% replacement. And then 2023 was almost 100% replacement. So all of the queens that we were selecting actively, basically they were in charge of the population. Same. And then we had the percent of recapped cells. So the number of recapped cells in these samples that I was taking, you had some colonies in 2020 that had absolutely no expression of recapping whatsoever. And even in 2021, when you had the brood infestation rates so high, you still had colonies that were not performing this trait. But in 2022, not a single colony actually didn't have recapping. So everything did. And then it's just kind of increased from there. And the recapping accuracy, again, you had some colonies that had a 0% accuracy. So they were recapping stuff, but they just missed all the time. And then in 2023, you had actually colonies that whatever their accuracy was, they still got some mites. And then some colonies were crazy with 100% accuracy, 75% accuracy, which was very, very high. Just to compare, Tadia's colonies have about a 60% accuracy rate. So those were the big wig survivors. Right. How much time do we have left? I'm just going to check. Oh, great. We're fine. Cool. So what worked and what didn't? We did? Yeah. Right. No. We're going to start with what was learned. So bees are not defenseless. We learned about 10 years ago that these bees definitely have traits that we can use to breed for resistance. Surviving colonies in Norway at least kept mite levels low for the entire time. There was really no period where they got out of hand. Could they get out of hand at one point? Probably if there were enough stressors piled onto the bees, they could lose control of the mites. That's definitely something that could happen. Most surviving colonies, kept the mites between under 10 mites a day on the boards in Norway. And again, this seems very, very high, but in Norway, the colony density is much lower than a lot of other areas. So you have a much lower horizontal mite spread. And I think horizontal mite spread is probably one of the large drivers, especially between late summer, early autumn, going to be one of the largest drivers of mite population growth. We don't have a lot of tests in place to actually tease that apart. I think just a few studies have been done. So we're still working on that. So recapping is present in all investigated resistant populations, but the behavior is really hard to pin down and quantify specifically because it changes with the level of threat. And generally, good survivors don't have a high level of threat. So it's hard to monitor. Right. So what worked and what didn't? Selecting based on bottom board counts really does seem to change the level of hygienic behaviors, and it's definitely changed the level of infestation in the brood. I would have loved to have collected more years, a couple more years of data. You always want those few years more, just to really find irrefutable evidence, but we didn't have that. So I'm pretty sure that what we did was directly impacting the infestation rates. Of course, it could change. It could go back up. Um, and we'll have to figure out exactly why, whether it's environmental, whether we're losing the traits. I feel like selecting for Varroa resistance is kind of an active, ongoing thing. You can't just select for 5 years and be like, okay, I'm done. I've got the surviving bees. No, you'll constantly have to keep putting that pressure on, or you might lose the traits that you really desire. Interesting, James, when you said IPM to you means just switching out chemicals, because in Norway and I think in most of Europe too, IPM usually means a bunch of different strategies and treating with chemicals as the last possible resort. So that's what I mean when I say IPM is, you can use treatments to basically help the colonies that you know are going to be kicked out of your breeding program, just so that they don't die, and then you can requeen them later. So using IPM to me means managing the mites so they don't get out of control and you don't drown your survivors so that they don't have a chance. But it also means keeping your stock alive, so not letting them die and then requeening them later, which is a good way to save money in bees and probably effort as well. So that's what I mean when I say IPM is, you use a bunch of different strategies, you select, but then you also make sure that your colonies aren't dying because of the selection, that you are actively cutting queens out of the breeding program yourself, and not letting them die. Um, would this have happened faster if we managed drones? Probably. As we saw in some of the other talks, that the artificial insemination or the instrumental insemination produced some of the best results there. This particular system was completely open maiden. There was absolutely no control of drones whatsoever. It probably would have happened fast. Why we didn't, one is because the beekeepers didn't have the means to do instrumental insemination at that time there. But also, one of the things that I wanted to keep was the genetic diversity of the stock. So I wanted the queens to have their choice in mates. I wanted the competition to happen naturally. And also, it's probably the most common way that apiaries are managed in Norway. And I wanted to do what the beekeepers were going to do. And then take data to see if that was a viable strategy for Varroa resistance breeding. Right. So that's basically the talk. What I'm working on now actually is a book, and I'm actually going to try and kickstart it. So I'm working on this book with Stephen Martin and with Marla Spivak. And what this book is going to be about is basically collecting all of these cases that we know of people breeding for Varroa resistance, and then profiling them, do 4 massive case studies, and then basically run almost like a guide of what they did, all of these cases, what worked, what didn't work, what were the similarities, what were the environmental challenges and differences between these cases, and just kind of draw conclusions to see if we can't kind of collect all of these instances and come up with a baseline guide for people who want to start breeding for treatment-free bees or breeding for Varroa-resistant bees. So we've got a webpage, resistingvarroa.com. And it's an official kind of pre-launch webpage to the Kickstarter before the Kickstarter goes live, and the Kickstarter is going to launch on March 10th. There's a subscription form that you can fill out to get notifications. So when the project goes live, you'll be the first to know. And we got a bunch of rewards here. So subscribers, so backers will get a copy of the book in one of the first tiers. And you can also get this really cool splash art that we use for our campaign, done by myself. Actually, I did the art for the book as well, and I designed the toy. There's a teddy bee that you can purchase as well as a reward for backing the project. And that is the end of my talk. So thank you very much for listening, and I'm happy to take questions now.
Thanks, Dr. Adi. So just a clarifying question. I may have broad brushed a little bit on the IPM, but there is a good majority of I'd say IPM education where the thrust of it is just make sure you rotate your chemicals. That's good integrated pest management. And so the education component with the mechanical means gets left out of a lot of those conversations, unfortunately. But there are some that hold strong and do teach it accurately, but I'd say the majority is just rotate your chemicals.
Okay.
So we do have some questions queued up.
All right.
In Norway, how many colonies do commercial versus hobby and sideline beekeepers run, and what's the level of migratory beekeeping in Norway?
Right. Cool question. Again, very small. So the largest beekeeper that is in Norway had 1,500 hives last I checked. The average commercial operation is 400 or 500. Hobbyists keep anywhere between, 2, 5, 10, 15. The queen breeder that we were working with was actually a hobbyist in Herdal, and he had 60 hives, give or take. And then we bred a bunch of queens, not just on his stock, but basically any queen that was in the project that scored high on the algorithm that I designed. We bred from, so the population was about 100 colonies. So yeah, migratory beekeeping. Yes, that was the part. So migratory beekeeping is not done in the same way or for the same reasons as the States mostly. It's mostly heather honey migration. So after the summer crop, beekeepers will move their hives onto heather fields to collect the heather crop. And that's the last crop that they collect. So that's what goes on in Norway. You do have some pollinator beekeepers, but they aren't very many because there's not a lot of crops that require pollination in Norway.
Another question from Serena, who asked the previous one. What are the viruses that you tend to find in Norway? Are the ratio of the various DWV types also there?
Yeah, so they are there. Pretty much most of the viruses that you're going to find in commercial honeybee operations generally you're going to find there. The interesting thing is I put out a paper not too long ago, I think about a year, looking at difform wing virus and the rates of these viruses in surviving versus non-resistant bees, and the rates were lower. So the prevalence and the quantity of the number of the viruses were lower in bees that could resist Varroa, which makes sense.
Okay, so in Dr. Trainer's presentation, she distinguished MNR, previously SMR. I'm gonna ruffle some feathers with that, and VSH. At the conclusion, she stated MNR is not a good selection criteria, so VSH is probably better. if I understood correctly, M&R is measuring the number of unreproductive mites, while VSH is measuring the removal of productive mites. I was too slow to get the clarifying question in the chat.
No worries. I mean, I can speak to it a little bit. So I actually found similar results in the MNR department, in that when I was looking at reproductive rates just straight up in these colonies, I wasn't finding a major difference between even between Tadeusz bees and the controls, but also the number of cells that I was looking at, infested cells in Tadeusz colonies, were like to 5 in like 500 cells, whereas, there was about 30% infestation rates in some of the control colonies. So that would definitely affect the averages that you're looking at. But also, if I was talking about this behavior being turned on and off, if there is no trigger, if there is no threat, then the behavior might be low enough so that the few mites that there are existing in the colony could reproduce as normal, and you wouldn't see a difference. So because of the variability, I completely agree with Dr. Trainer, is that's a very difficult way to, and it's not a reliable way to assess the performance of your stock.
Since horizontal mite transfer can be high, how far would you think a bee yard should be apart to negate horizontal drift?
That depends on so many environmental factors. Where are the winds blowing normally where you guys are? Is there a mountain in the way? The geological formations and stuff play a very big role. Where's the forage? Is the forage, right in between these apiaries? Is there a reason they could be transferring back and forth? We have numbers, but it really depends on the location. We say that I think 1 to 1.5 kilometers apart is enough to really prevent a large amount of transfer. But then you got to think about robbing as well. If there's a weak hive and it's detected by your stronger hives, even if it's, up to 3, 4 kilometers away, they'll get it if there's nothing else in the area. And robbing is where you can have a very high mite transfer.
If the recapping was not removing the pupa from the cells, but you determined that there was a level of accuracy, what exactly were the bees doing accurately? Were they removing or disrupting the
Just
mites?
recapping. Yeah, so I mean, I have a theory, and of course it's really hard to get solid data, because if you're looking at just the bees doing the recapping, they're making selections, right? So they're choosing to recap some cells and ignoring other cells. That's a bias introduced. So when you're just looking at what the bees are doing, it's very hard to reliably relate those reproductive rates to the actual effect of recapping versus if they're going to ignore a cell because it's non-reproductive, for
Hmm.
example. So all we know is that there's very high levels of recapping in bees that can resist Varroa, and I did not find brood removal. Not that it's not there, but when I was looking for it, I did not find it. Um, what I theorize is that the juvenile mites are extremely sensitive to changes in temperature and humidity, and that simply opening the cell at critical periods of the mite's reproduction could actually hinder or destroy some of these cells. A lot of the eggs are actually laid fairly close to the top of the cell, and the bees could unintentionally destroy them. Or the air could be dry enough to desiccate the eggs and young protonymphs. So that could be affecting the mites. They could also be interfering in mating. Like I said, the sperm transfer is very conservative in Varroa, so if you mess up the mating, they might not get another shot later on. Um, and as to why I didn't see brood removal, the only theory that I can throw at you is the higher north you get, probably the more valuable winter bees and brood are, so that you don't want to be throwing them out, even if it's for, Varroa control. And recapping may be kind of a secondary but more viable option in colder climates where you need to have that you need to be highly conservative about your winter brood. Whereas in more southern climates with shorter winters, shorter brood breaks, the bees don't have to survive for as long. Maybe brood removal is a much more viable strategy, which is why we're seeing it in all these other populations. Or maybe introducing strange brood just didn't trigger the removal. That's
That
also a
sounds like
possibility.
a budding study.
Yep, there's a lot of work.
Yeah, it's never ending. Um, Do you think that, and kind of to piggyback on that, do you think that uncap recap without brood or pupa removal lowers mite reproduction?
I think it could. I started to do a study during my postdoc of myself, basically looking at these colonies that had relatively no recapping naturally, and then just recapping the cells myself, or uncapping them and letting the bees recap. I started to see a decline in male mite presence. And an effect on deuterymphs, female deuterymphs as well, but I couldn't collect enough data for statistical significance because it was a very labor-intensive project. Um, and they ended up removing most of the test cells because I'd been tampering with them probably and opening the colonies so often. There was a visible change in colony behavior by the end of the experimental period. So I'm probably going to need a lot more colonies and a lot more time to get that data out in a good capacity.
So this is another one of those common overlaps with terms. Is uncapping and balding the same behavior? I was under the impression they do not recap when you see balding, and do they eventually pull out all the exposed pupa?
So balding can happen for various different reasons. And sometimes when you see balding, especially in large patches on a frame, it means that your bee space, so the space between the frames is too narrow. And that's a behavior that some colonies do if they don't have enough bee space. So that is not related to uncapping and recapping for hygienic purposes. That is something different. Um, I can't think off the top of my head any other reasons why you would have mass amounts of bald brood. Um, but for at least those two examples, they are unrelated.
So there is a beekeeper in the state of Vermont named Kirk Webster. Are you familiar with Kirk?
Yeah, I
Okay.
know Kirk. He's a good friend. Sorry,
So
there's a really good comment in the chat. Wax moth also causes balding, and that actually recapping is so wax moth is a target for high levels of recapping as well. Recapping is not just a Varroa thing. It actually originally wasn't because there was originally no Varroa, so it was used for a lot of other pests and pathogens. So sorry, Kirk, go.
no, that's all right. Yeah, no, there's a lot of overlap there. So a keen eye will help the beekeeper distinguish what's happening if it is actually some sort of a resistance mechanism or if it's wax moth. My personal leaning is if it's wax moth, I'm gonna likely be looking at a weaker colony that's not strong enough to be fighting them off individually. So it's more likely that it is wax moth. But if it's a very strong colony, low likelihood. Um,
Yeah,
yeah,
you're
so
definitely going to see wax moth if you have wax moth.
right. And so yeah, Kirk was recommended by by one of the attendees here that might be a good candidate for your project.
Yeah, definitely. Actually, I've been talking to Kirk. I wanted because I'm in Montreal currently. I want to drive out to him and see if I can take some samples and run the tests just to see if I can find recapping in his stock too.
I'm sure he would be happy with that. The
Yeah, I definitely have to
question,
visit.
yes, you have to visit. He's not a virtual person. Um, could we mimic uncapping, recapping by using a capping scratcher or lightly opening brood cells? Would it at least create one generation of mites that weren't able to reproduce? And could this possibly train bees to engage in the behavior? I think this is
So
hinting
that's
at
the experiment that I tried to do. I couldn't get enough data, and it seemed to be working. Um, to the question of can you train bees to do this? Probably a bee's lifetime, especially in summer, of 30 days is probably not enough to transfer any sort of knowledge to the colony. And we actually did a little bit of an experiment on can this behavior be behaviorally transferred or is it mostly genetic? The results haven't been published, but from what I can see, genetics tend to play the more dominant role in, the expression of the trait. So if you have a surviving queen or queen from a good surviving stock, then chances are you're going to see a lot of recapping. Whereas, if you just give hygienic workers to a susceptible colony, as soon as those workers die off, you're probably going to see much less hygienic behavior.
Hmm. Is the methodology of the Herdal project published?
Not yet. No, in the works. Actually, a lot of the data I showed you was not published yet, so
Okay.
hush. You got
Yeah.
a special treat.
It's all private. It's all quiet.
It's all
Um,
recorded anyway, but it's all right.
when measuring the uncap recap accuracy, how do you know if the bees uncapped a mite infested cell? The mite moved out and the bees recapped it afterward?
Generally, once a mite has committed to egg laying, she does not leave the cell, not even if the cell has been disturbed.
Hmm.
So yeah, and if the mite has left, then you generally do see evidence. You see the fecal pile on the side of the colony. It was very, very rare that I saw evidence of mites with no foundress in. So there was almost always a foundress. We're
Uh,
talking maybe 1 to 2 cases in about 500 cells.
hmm. Do you have a personal website that you are maybe sharing when your publications go live?
I do not. That's a good idea. I should probably put one in the works. That's I'm still kind of building my career. It feels like I just finished my PhD. I'm 7 years out of my PhD. It feels like I just finished.
Well,
So
you've done a
there's
lot
so
of
much
good
work
work
to do.
since you started. It's definitely
Thank
a
you.
growing corpus, and many of us are very familiar with your projects. So I'm grateful.
I'm
Um,
glad to be of service
yes, and so you are in Montreal now. And what are some,
currently.
besides the Kickstarter and the book? What are some other projects or things that you're looking to embark on here?
Well, finishing up data, and I am currently in transition, so I am going to be looking for a new position, hopefully in a assistant professorship or associate professorship at a university. There's a few opportunities, so I'll see what I get.
Hopefully, we get to see you in Detroit sometime, eh?
I hope so.
Well, thank you very much for your time and your flexibility with the scheduling and the presentation. It's been wonderful. We're glad that we got to see some of this top secret stuff.
My pleasure.
All right.
Actually, I'll put the link to the website in the chat. Do you mind?
No,
Is that
yeah,
all
go
right?
for it. Yep, 100%. All yours.
All right. And with that, you can definitely move on to your next speaker.
All right. See ya.